Hello, thank you for joining us today. I'm Sonjelle Shilton, the Deputy Head of HCV Access here at FIND. FIND is the Foundation for Innovative New Diagnostics. And we've teamed up with the World Hepatitis Alliance to create a series of webinars on hepatitis diagnostics basics. We hope that this information will be useful to you as you work to increase hepatitis testing, care, and access. The first webinar today will focus on hepatitis C diagnostic basics. It's intended to give you an overview of core concepts regarding diagnostics and specifically hepatitis C diagnostics. So a little background about FIND and why I am the one here talking to you today on this. We're a global nonprofit and we drive diagnostic innovation to combat major diseases affecting the world's poorest populations. We have offices in Geneva, Vietnam, India, Kenya, and South Africa. And currently, we work on a portfolio that includes hepatitis and HIV, antimicrobial resistance, malaria and fever, neglected tropical diseases, pandemic preparedness, and tuberculosis. So to start off with the very basics, what is an in vitro diagnostic, or IVD? So in vitro diagnostics are tests done on samples such as blood or tissues that have been taken from the human body. In vitro diagnostics can detect diseases or other conditions that can be used to monitor a person's overall health or to help cure, treat, and prevent diseases. So in a nutshell, in vitro means happening outside the body. So this is any time you take a sample such as blood, saliva, hair, tissues, you take it and you perform a test in another area, usually a laboratory or a device. So when we talk about diagnostics, you might hear a few terms often bandied around. These are sensitivity and specificity. So sensitivity refers to what percentage of the time a test will correctly diagnose somebody with the disease who has the disease. Specificity is the percentage of time a test will correctly diagnose a person as not having the disease when they actually do not have it. So I like this picture here. In the picture you can see a doctor with a man, and the doctor is telling the man that he is pregnant. You can see another doctor with a woman who is obviously pregnant, and the doctor is telling the woman, "You are not pregnant." So when the doctor is telling the woman she is not pregnant, when she obviously is, that's an example of something that is not sensitive. And when the doctor is telling the man that he's pregnant when he obviously is not, that's an example of a test that's not specific. So when we talk about diagnostics, we want to ensure that the diagnostics we're using are quality. And one of the ways that you can do that is stringent regulatory authority. So why do we need quality diagnostics? Well, there are several components that go into it, but you want to ensure that the diagnostic test you're using is giving you correct results. For today's webinar, we'll focus on one aspect of the quality chain, and that is stringent regulatory authority or WHO Prequalification. Stringent regulatory authority is a regulatory body that ensures that a diagnostic is quality assured. Basically, does the diagnostic test do what it says it will do? Will it correctly identify a specific disease in the sensitivity and specificity ranges that the manufacturer says it does? And also, stringent regulatory authority ensures that the manufacturing facility and process is uniform. This is important because you want to be able to be assured that every lot or batch of a diagnostic test you get will perform in the same manner. You don't want to be in a situation where you order a bunch of tests, they show up, you use them, they provide high quality results. You order that same test again, it shows up, but this time it's not providing you with those quality results. So stringent regulatory authorities help to independently validate and verify that a test does what it says it will do, and that the manufacturing processes are quality and uniform, so you can be assured that each batch of tests you get will provide you with the same results. There are different types of regulatory authorities. The first one is global. Now this would be WHO Prequalification, or oftentimes WHO PQ it's called, or the Expert Review Panel for Diagnostics, which is oftentimes called ERPD. And then you have country or regional specific stringent regulatory authorities. On the slide deck here, which we'll be sharing as a resource, so you can reference this in your own time, we've listed just a few of the country or regional specific stringent regulatory authorities. There should be hyperlinks here, so you can click on each of the underlined to learn more about each one that's listed. So in Australia, there's the Therapeutic Goods Administration. In the European Union, there's what we call CE marking. In Japan, there's the Pharmaceutical and Medical Devices Agency. And in the United States, there's the US Food and Drug Administration, which is often referred to as FDA. So on these links here, you can click to see the information about these different country or regional stringent regulatory authorities. So let's talk a little bit more about WHO prequalification. This may be one that could be relevant to you in your setting. So why is WHO prequalification important? Well, not only does it help to ensure the quality of diagnostic, but if a diagnostic has WHO prequalification, then that specific make and model of the diagnostic test can be procured through UN and Global Fund. So we've listed here some sources. The first one is the overall information about WHO PQ. The second is where you can look to check to see if a specific test has achieved WHO prequalification. And the third is a source that if you're very interested in this and want to learn more, understand how the process works for WHO prequalification, you can click on that third link. To talk a little bit about the Expert Review Panel for Diagnostics. Now, ERPD is hosted by WHO, and it consists of a group of experts that independently review diagnostics that have not yet gone through WHO PQ or are in the process of going through WHO PQ. Diagnostics that are reviewed by the ERPD and approved are eligible to be procured by Global Fund. This is important because the WHO prequalification process is very stringent and sometimes can take some time, whereas the ERPD harnesses the knowledge of the technical experts and is able to give a judgment that may allow a diagnostic to be procured while it is still going through the WHO PQ process. The two links that we've listed here, the first is overall information about the ERPD process, and the second is a link where you can look up what diagnostics are currently having ERPD approval. So context is important when we're talking about stringent regulatory authority. Each country has its own landscape when it comes to diagnostic regulations. Some countries require in-country studies to be done on the diagnostics before they are allowed to be registered in the country. And other countries accept tests if they have WHO prequalification or another stringent regulatory authority already. So it's a complex landscape, and we recommend to you that you look to see what resources are available so you can find out the regulatory landscape in your country. Some countries list online the diagnostics that are registered, such as India, while as other countries, it may be useful for you to contact your Ministry of Health or your WHO country office to see if they can point you to information about what the diagnostic registration process is and what tests are currently registered for hepatitis C diagnostics in your country. So now that we've covered those very basics, let's move on to the WHO hepatitis C testing algorithm. To understand the WHO testing algorithm, it's important to understand the progression of hepatitis C illness. So when a person is infected with hepatitis C, it usually starts off with an acute infection. This is a quick onset infection. Of the people who have acute infection, about 20% of those, their bodies will spontaneously cure the illness, meaning that their immune system will fight off against the virus and it will not progress to chronic inflammation of the liver. However, about 80% of people who get acute infection will progress to chronic inflammation and infection of the liver. It's called hepatitis C viremia, and that can lead to fibrosis, cirrhosis, and liver cancer. We contextualize this here for you because the hepatitis C diagnostic algorithm is a little bit complicated. It requires more than just one test to understand if somebody needs to be treated for hepatitis C. The reason for that is that 20% of people who spontaneously clear the infection, they will test positive on the first hepatitis C diagnostic test, but they will test negative on the second test for viremia, and they will not need treatment. However, around the 80% who will have acute infection and then progress to chronic infection, they will test positive on the first hepatitis C test, as well as positive on the second test for hepatitis viremia, and they will require treatment. So with that context, let's move on to the WHO algorithm. So the WHO hepatitis testing guidelines were issued in 2017. And we've provided a link on the top of the slide where you can go to see the information that WHO has produced, as well as the testing guidelines. These guidelines were compiled with a strong evidence base, using all available evidence to understand the most optimal testing algorithm for hepatitis C. We can see that it's divided into 4 parts. The first part is that single test for screening. So that's a single test to understand if someone has been exposed or had acute hepatitis. The second is a prompt test to understand if the person who has been exposed is in fact viremic and thus requires treatment. The third test is a test to determine the liver stage. So we want to understand how damaged the liver is at this point. Once you have those 3 informations, those 3 tests, then you can treat all with pan-genotypic regimens. And step number 4 is 1 test of cure at 12 weeks after completion of treatment. This test is done to determine if the treatment was effective and the person is thus cured of hepatitis C. So looking at the first step in the algorithm, serological testing for hepatitis C. So what are serological assays or serological tests? Well, they're tests that screens for hepatitis C. Serological assays are tests that detect the presence of either antigens or antibodies, typically in serum or plasma, but also capillary and venous blood and oral fluid. These tests include rapid diagnostic tests, or RDTs, which are immunoassays that detect antigen or antibodies and can give a result in less than 30 minutes. And I have here an example of a rapid diagnostic test, so you can get an understanding of the size. It's quite small. This one is in a cassette, so it has a plastic casing around it. Now what you would do is you would put the sample type, so be it plasma, serum, capillary blood, in this circle here. Usually apply a buffer, wait the specified time as per the manufacturer's instruction, and then the results would come down here, and you would read the results of the test. The other way that you can do serological testing is EIAs, and these are enzyme immunoassays that detect antigens or antibodies. Now these ones are usually larger machines wherein multiple samples would go in and be read for results. So you can think about hepatitis testing in 2 contexts. You have centralized settings and decentralized settings. So when we say centralized settings, we're usually meaning larger health facilities, hospitals, tertiary facilities, places where you have a lot of infrastructure and space, as well as a lot of manpower. In these settings, you can screen for hepatitis C using EIA or CIA, which are again these large platform machines. These require a well-equipped lab with good constant electricity, a qualified lab technician, and these are done on plasma and serum, and they usually take around 2 or more hours to complete. Now, usually when you're doing EIA or CIA testing, it requires that you batch samples. This means that you have a large number of samples collected, and once you collect all the samples from different people, you run all of those samples at one time. Now, in decentralized settings, this could be something more like a primary healthcare clinic or even community-based testing. These settings, you can use the RDT blood-based, or for hepatitis C, there is an oral fluid-based RDT. And we have a picture on the slide so you can see what that looks like. Now, in these settings, you still do require a trained healthcare worker to administer the test. And you can use capillary blood, oral fluid, and the turnaround time to results is in between 5 and 20 minutes. Now, there's an interesting thing that can be done where you can collect the samples in the decentralized setting using dried blood spot. So this is usually capillary blood from a finger, which is put on a special piece of paper, collected at the decentralized settings, and then sent to the centralized settings to be tested using the EIA or CIA. Currently, DBS, dried blood spot, for screening is used off-label. That means that the manufacturer protocols have not yet been validated. What's needed to validate these are a series of evaluations and trials, and I'll talk a little bit more about that in a few slides down the road. So this next slide here, this is mainly for your take-home reference and reading. And what this lists is the hepatitis C antibody RDTs that are currently WHO PQ'd or ERPD. Now, this list is evolving as WHO PQ and ERPD review new technologies. So we always recommend that you check back in those links in the earlier slide where you can see the current tests that are WHO PQ'd and ERPD. But here we have the table which shows the product name, the manufacturer, the performance, the sample type used, if it is PQ'd, the stringent regulatory authority that it has, as well as the list price in US dollars. When we talk about price of diagnostics, it's always a little bit tricky because prices can vary a lot. It can depend on the volume that you're ordering, the region of the world you're ordering from. So these list price here are to give you an indicative understanding of the prices, but they may vary in your region. The second slide is the hepatitis C EIA tests that are WHO pre-qualified. Again, product name, manufacturer, sample type, stringent regulatory authority. On this table here, you'll see a technology called the Architect HCV antigen assay. Now, this assay can be used for confirmation of viremia as well as screening. And I'll talk more about that when we get to the step 2 confirmation of viremia section. So continuing on with serology, or the screening test, I'd like to talk about some interesting developments that are coming up. One of them is self-testing. You may be familiar with self-testing from the world of HIV, wherein self-testing is now becoming a more accepted technology. In order for self-testing to be used for HIV, there had to be a series of studies and trials done to understand if it was feasible, acceptable, to understand if people who did self-testing were being linked to care in the proper way. These same studies need to happen for hepatitis C self-testing because currently there is very limited data available on hepatitis C self-testing. There's been a few studies that have shown that it could be acceptable, but there are multiple concerns associated with hepatitis C self-testing, primarily around access to confirmatory testing and further linkage to care. In the second study that we've got listed here, participants found that the testing procedure was easy, but they did report some difficulty in interpreting test results. And it's important to note that this second study here, This was using self-testing, but guided by a healthcare worker. So a healthcare worker was with the person and sitting next to them while they performed the self-test. So in order to add to the evidence base to understand if and how to use self-testing for hepatitis C, FIND is conducting a pilot feasibility study. And so we are working in several countries in different geographic regions to understand the performance, the acceptability of the OraSure HCV rapid diagnostic test, which has been adapted by the manufacturer for self-testing. It's a research use only at this point. We're working in Egypt, China, Kenya, Georgia, and Vietnam, amongst a variety of different population types. So general population, men who have sex with men, people who inject drugs, and we hope to correlate these data to better understand the next steps on what more research is needed to advance hepatitis C self-testing. Another interesting advance is combo testing. So the fancy name for combo testing is multiplex serological testing. And these are rapid diagnostic tests wherein you can test for multiple diseases on one strip using one sample. So this table on this slide here shows some of the tests that are currently available for combination testing. And usually you'll see that they detect for hepatitis B, hepatitis C, and HIV. Now, field validation is needed to assess the diagnostic accuracy of these tests. And what that means is that independent organizations, not the manufacturer, but rather independent organizations, need to do studies and trials on these tests to understand the sensitivity and specificity, the performance of the test among all three diseases that they test for. Additionally, further work needs to be done to understand, are these tests cost effective? What is the impact of these tests? Are people being linked to care in the proper way if the tests do diagnose one or more of the diseases? So work on this is ongoing, and we hope that within the next coming time, we'll be able to understand better how to most effectively use the combination tests. So now we're on to step 2 of the diagnostic algorithm, diagnosis of hepatitis C virus, confirmation of active infection, or confirmation of viremia. So as we see from the WHO algorithm, this is step 2, and they say prompt or reflex HCV RNA or HCV core antigen testing. So just as we had in the screening section, you can think about confirmation of viremia in the same way, centralized settings and decentralized settings. In centralized settings, you can use the HCV core antigen and the HCV RNA test. So these again are large platforms wherein you need a well-equipped lab, a qualified lab technician, and the specimen type is plasma or serum. The turnaround time can be around 5 hours. With these large machines, normally, not in all cases, but normally you have to do batch testing. Again, that means collecting a large number of samples, waiting until you have a full plate, as they call it, running the machine after it's full. In decentralized settings, we have the option of HCV RNA testing. There are 2 technologies currently available on the market which have stringent regulatory authority for hepatitis C testing, and that is the Gene Expert and the Gene Drive. So these decentralized settings can be district hospitals, lower level, but still needing a trained healthcare worker. The specimen type is capillary blood or plasma, and the turnaround time to results is around 60 to 90 minutes. In development is a core antigen RDT for hepatitis C. However, that work is still in the development phase and is not yet on the market. So just as we had for the screening section, you have the option of using dried blood spots for HCV RNA. You can collect the sample in a decentralized setting, such as a community outreach, primary health care, or district hospital. You can collect the blood there, send it on the dried blood spot card to the centralized setting to be run on one of those larger platforms. So this next slide here is a busy slide, and this is just to show you a few of the large throughput platforms that can perform hepatitis C RNA testing. And then we have listed here 2 platforms of Roche, one of Abbott, and Hologic. We've also listed the assays. That's the different types of tests it can do. HIV, HPV, HCV, HBV, and TB in some cases. The sample types for HIV and HCV, the space requirements, human resources, and workflow. So a little bit more on dried blood spot. So in order to ensure that the manufacturer protocol for dried blood spot collection and testing is accurate and validated, FIND is conducting a multicenter diagnostic accuracy study. And we are looking at the performance of DBS on Abbott M2000, Roche 48 and 6800, and the Hologic Panther. We're doing this study in Georgia, Greece, Cameroon, Rwanda, and Australia. And we are finishing it very soon. And we hope to be able to provide the information and evidence to the manufacturers so they can submit for stringent regulatory authority for this DBS next year, in 2020. The advantage of DBS is it allows you to take the sample collection very far down the health chain. So you can collect the sample in community outreach, community testing, and then send it to the centralized labs. It can help overcome some shortcomings if maybe the confirmation testing isn't available in your area, perhaps rural areas, areas with less infrastructure, etc. Another way that you can do confirmation of hepatitis C viremia is point of care, sometimes called POC or POC, and near point of care. So, as we already mentioned, there are 2 platforms that have stringent regulatory authority for hepatitis C viremia testing. And they're listed here. We have the Expert HCV viral load assay, which uses plasma, the Expert HCV finger stick assay, which uses capillary blood, and the Gene Drive HCV assay. Now, you'll see on this table a lot of information about the platforms. One of the things it says is sample preparation. For the Expert, it says integrated, and for the Gene Drive, it says off-board. Now, what that means is if you use the Expert assay, you can put the sample into the cartridge and then put it into the machine to run. However, with the Gene Drive assay, there are a few pipetting steps that are needed from the sample to the sample preparation to putting it in the machine to be run. So next we have some up-and-coming technologies. One of these is the MolBio TruePrep. So MolBio is a commercially available platform which is used for TB testing in India. It has internal rechargeable batteries. This could allow for implementation in facilities without electricity support. And currently they are finalizing the development of the hepatitis C assay for this platform. FIND is preparing a multicenter prospective diagnostic accuracy study on this HCV assay. And so we hope to evaluate the performance of the assay and understand the sensitivity and specificity to provide evidence to MolBio to then submit this to stringent regulatory authority. Another platform that's up and coming is Blink. Now, this platform is not commercially available yet. It is still under development. It is intended to be a multi-purpose platform which can detect a wide variety of things, and Blink is intending to develop many different assays or tests on this platform. You can see the information about the prototype HCV assay with a relatively quick turnaround time and high sensitivity and specificity. Now, Blink is continuing to develop the HCV assay, and their target to complete product development, validation, and launch is Q1 2021. So to talk a little bit about the core antigen rapid diagnostic test that I mentioned earlier. So the core antigen rapid diagnostic test, the intention of this test is to be able to bring confirmation down very close in the community level. So the hope is that you would be able to give a serological screening rapid diagnostic test. And if that person screens serologically positive, immediately you could give another rapid diagnostic test for a core antigen to see if that person is viremic. And it would be able to give results very quickly. So it has the advantages of being able to bring the diagnostic very far down into the health system. However, because of the nature of the technology and because it would hopefully be in a kind of cartridge like this, the core antigen RDT would only have a sensitivity of 75 to 85%. That means that some people that are viremic would show up negative on the test. And so they would thus still have to go for the centralized confirmation using an RNA or a more sensitive core antigen technology. So that's what this algorithm is trying to represent down here on this slide. The good thing is you would catch the majority of people who need to be on treatment right away in a low-level setting, but there still would be a few people that would need to go to the higher level setting to have the more sensitive viremia testing done. So now we're moving on to stage 3 of the algorithm, liver staging. So you need to assess and triage. You need to conduct liver staging tests to understand how much damage the liver has. This is important clinically because there may be other health concerns that need to be addressed with the liver if it's very damaged. So even if you cure the hepatitis C, the doctors may need to manage the patient for other aspects around liver disease. So just to list some existing liver staging options. We have the biochem. And so what this is basic blood tests that are used to determine either APRI or FIB4. And these are formulas and ratios taking into account other biochemical readings, sometimes with age, and then that can give you information as to the liver scoring of a person. The advantages of biochem is that these results are usually attained from tests that are already existing in district hospital or tertiary hospital facilities. They're usually already found in those facilities and they're usually relatively inexpensive. Another option for liver staging is FibroScan, and this is a machine that can provide liver staging results. However, the machine is quite expensive and it requires a trained technician and is not widely available in all countries or contexts. Alternatively, you could use ultrasound. This can also be used to provide liver staging results. So while often ultrasound machines are already in district or tertiary hospitals, it does require a trained technician. And we're seeing in some of our work that the wait time for ultrasound for liver staging can be quite long. This is because other patients, such as pregnant women, may be prioritized for ultrasound results. So there are a few interesting technologies in the development for liver staging. One of them is done by the Burnett Institute and Nanjing Biopoint, and they are working on developing an RDT that would be used for liver staging. They have already started some trials on their RDT and hope to be able to provide more information soon as to the development and how that's going. So now the last stage in the algorithm is one test of cure at 12 weeks after completion of treatment. So currently the recommendation from WHO is that this be done using an HCV RNA test. That can help determine if the person has been cured of the disease and no longer has any viruses that are detectable left in their system. So this last slide here is just a kind of overview of the product pipeline that shows you what products are up and have come out and are up and coming for hepatitis C. And the last slide is the thank you slide. So thank you very much for staying with us. I was trying to get this in under 30 minutes. I see I'm just at 36 minutes, so pretty close. We'll be sharing these slides with you so you can reference them. And we'd be happy to answer any questions that you have about diagnostic technologies. The next video that we'll be working on is best uses and practices for hepatitis C diagnostics. So that is taking those, what are the best ways that people have taken the available diagnostic technology out there and use them to ensure high quality linkage to care, from screening to confirmation to treatment. So we do hope that you'll join us next time, and thank you for joining us today.